Welcome to MathGPT. Today we're going to talk about the NMDA receptor, a special glutamate ionotropic receptor. Our goal is to understand why the NMDA receptor conducts very little current at negative membrane potentials, but large current at more positive potentials. We'll use a clear visual model and then select the correct mechanism. Here are the choices. We'll read them slowly, and then we'll analyze the receptor step by step. Let's build an intuitive diagram. This rectangle is the cell membrane. The top is the outside of the cell, and the bottom is the inside. In the center is an NMDA receptor channel. Now, let's show a negative membrane potential. That means the inside is more negative than the outside. We'll draw negative signs inside. At negative potentials, extracellular magnesium ions are pulled into the pore, and they block the channel. When magnesium sits in the pore, ions like sodium or calcium cannot pass, so the current is very small. This is why at negative membrane potentials, the NMDA receptor conducts little current. Now, imagine the membrane depolarizes, which means the inside becomes less negative or even positive. At positive potentials, the magnesium is pushed out of the pore. The channel unblocks and ions can flow through. This is the key idea. The NMDA receptor is voltage dependent because magnesium blocks it at negative potentials and depolarization ejects the magnesium. So the mechanism is a voltage-dependent magnesium block, not a special voltage sensor. Now, let's return to the answer choices. The correct choice is option A, blocked by extracellular magnesium. The other options describe calcium block or a voltage sensor segment, which are not the primary mechanism for NMDA receptors. Let's recap slowly. First, the NMDA receptor needs glutamate and glycine to open, but even when it is open, magnesium can sit in the pore. Second, at negative membrane potential, magnesium is pulled into the channel and blocks it, so very little current flows. Third, when the membrane depolarizes, magnesium is pushed out, the channel unblocks, and ions, including calcium, flow through. Therefore, the voltage-dependent magnesium block is the reason for the small current at negative potential and the large current at positive potential.